Hello everyone. Today I'm going to present the poster from our PBL group, PBL H27. First of all, I'm going to introduce the entire team. Our group consists of 11 people and is led by Ramadan. The rest of the team consists of me, Beckham, Michael, Hazwani, Batrisha, Cindy, Akilatul, Filzatun, Huda, Mary, and Scarlett. Mr. Razif is the facilitator for our group. Now, moving on to our poster. Angiosperms are from phylum Trachyphyta, divided into two classes, monocots and dicots. To define whether a species is monocotyledons or dicotyledons, the characteristics of the plant species are classified into respective categories, such as seeds, leaves, stems, flowers, and roots. This documentation aims to identify different species and habitats of angiosperms in Kuba National Park. Using field observation techniques, different species of angiosperms were identified and differentiated along the summit trail. From the trip, we have observed that there are 4 species of angiosperms present in Kuba National Park, which are Arundina graminifolia, Dilena subfruticosa, Zingiber zerumba, and Stasitafera jamaicensis. In conclusion, Arundina graminifolia was observed in high abundance at Kuba National Park, which may be due to favorably humid environment. Now, to provide some background on our research, Kuba National Park is one of the national parks in Sarawak which covers 2,230 hectares of mixed dipterocarp forest that is rich in palms, orchids, and pitcher plants. Angiosperms are the most diverse group of land plants, with 64 orders, 416 families, about 13,000 recognized genera, and 300,000 known species. Angiosperms can be divided into dicotyledons with 2 seed leaves and monocotyledons with 1 seed leaf. Angiosperms are vascular seed plants in which the ovule is fertilized in an enclosed hollow ovary for it to grow into a seed. The objective of this strip and documentation is to identify different species and habitat of angiosperms in Kuba National Park. Moving on to the methodology, our documentation was conducted in Kuba National Park, which is located 20 kilometers away from Kuching City in Sarawak. The documentation of angiosperms was conducted along the way to the summit of Mount Serapi. We used several ways to identify the angiosperms that could be found in the national park, which include reading materials, internet, and consulting our facilitator. Our trip to the national park discovered 4 species of angiosperms. The first is Arundina graminifolia, also known as bamboo orchids. It is a species of orchids and is the only species that belongs to the genus Arundina. This species was found at about 2,300 feet, growing along the summit trail. The flower had white petals with a ruffled labellum that is purple in color and a yellow center. The flower is succeeded by a ribbed seed capsule that contains the powdery seeds of the species, which would split open when ripe to release its seeds into the wind. The second species was Dilena subfruticosa. It is also known locally as Simpoh Air. They are large evergreen shrubs that can grow up to 6 meters tall with deep underground taproots. The species was found at about 2,300 feet along the summit trail. Once fertilized, the flower would rotate to face upwards. The fruit produced is pink and star-shaped, containing bright purple seeds, which would open up and are quickly eaten by birds and monkeys. The third species of angiosperms found was Zingiber zerumbat. It was found at up to 1,200 feet along the summit trail. It is red in color, cylindrical in shape, and have unbranched leaves with stems that are usually 30 to 45 centimeters long. Lastly, the last angiosperm species found in Kuba National Park is Stasitafera jamaicensis, also known as blue porterweed or blue snakeweed. It is usually found at a width of forests, roadsides, and disturbed sites. This species, which was found at 2,300 feet at Kuba National Park along the trail summit, was arranged on long, curved, relatively thick spikes at the top of the branches. These flowers are stalkless and either pale blue, blue, or mauve in color. In conclusion, 4 different species of angiosperms were observed and documented throughout the study area, which was along the Summit Trail in Kuba National Park. Arundina graminifolia was observed in high abundance in Kuba National Park compared to the other species of angiosperms, which may be due to the favorably humid environment. We would like to thank the Center for Pre-University Studies, University Malaysia Sarawak for the financial support, Sarawak Forestry for the visiting permit, and our biology lecturers for guidance and assistance. We are extremely grateful for the opportunity to experience research at Kuba National Park. That is all from our PBL group. Thank you very much for watching.